Hydrogen may be the tiniest atom on the periodic table, but in AP biology, it is the key to unlocking the mysteries of some of the most complicated processes that go on in cells. Hi, I'm Miss Parrott, and in today's video, we're going to be looking at why hydrogen matters in AP biology. You're going to see hydrogen in so many different places. It's one of the two elements that make up water. It's crucial for understanding acids, organic molecules, redox reactions, which are in AP Bio, not just AP Chem, proton gradients, photosynthesis, cellular respiration, and ATP production. So you're going to be seeing hydrogen and its pieces, parts all year long. In AP Bio, it's really important for us to imagine that a hydrogen atom is really one proton and one electron, and they can act independently of each other to drive cell processes. And by understanding this, we're going to be able to unlock the mysteries of not just cell processes, but of the flow of energy. So let's get into it. Like I said, we need to think of hydrogen as one proton and one electron. Now, a lot of times you're going to see this. So hydrogen is just H, and then the one proton is denoted as an H with a positive charge. That's just a proton. And then an electron is usually an E with a minus. Sometimes you see a proton as P plus in chemistry class, but in most biology textbooks, you're going to see a proton as a H plus. So if we're looking at a very simple atomic model, right, you have your proton in the nucleus, and then the electron is surrounding it in an electron cloud. So in this case, this makes a neutral hydrogen atom, and when we take the electron away, which often happens, electrons are always moving from place to place, we are left with just a proton and H plus. So it's often a misconception that H plus is a whole neutral hydrogen atom with an additional proton, but that's not right. So if that's in your brain, scratch it. This is going to be just a proton. Now, why does this matter? Because protons don't just hang out, they interact. And you're going to see these because, remember, biological systems are usually like roughly 60 to 80% water. And so water is a polar molecule, so it's going to be interacting with all kinds of things in our watery systems. It's going to interact with polar and charged molecules. And it's very important in making proton gradients and plays a huge part in energy transfer in cells. So first, let's look at electrons. So electrons are important because electron transfer between molecules is associated with energy transfer between molecules. And energy transfer is going to be a change in potential energy from one place to another place. Now, flashback to chemistry, oxidation and reduction. So, oxidation is when an atom loses, or molecule loses an electron. So, oil rig, OIL. Oxidation is losing electrons. That's what we're losing. And so if you lose a negative thing, then you become more positive, right? Reduction is gaining an electron. So if a molecule is reduced, it has gained an electron and is now become more negatively charged. And again, with oxidation and losing electrons also it's associated with the giving of potential energy, and reduction is gaining that. Okay, now you're going to see electrons being carried from one part of a cell process to another part of a cell process with molecules called electron carriers. these are going to show up in photosynthesis and cellular respiration. You've got NADH, NADPH, and FADH₂. So all these H's contain an electron. And so these molecules, the electrons that are part of those H's, are being carried from one place to another place. Also, you're going to see in photosynthesis, the final electron acceptor is oxygen. So an oxygen atom is going to accept electrons and then be able to form O2 and then be a byproduct of photosynthesis. So electrons, following the electrons helps us follow the energy. Now, protons are super important because they help cells build proton gradients. So now we're going to need to be pulling in some of our previous knowledge about diffusion and how molecules like to move from areas of high concentration to low concentration, and how if you have a separation in concentration, that's a gradient, right? It's higher in one place and lower in another place. So a proton gradient is an unequal distribution of protons. And so here's an example of one side of a membrane. Here we have a lipid bilayer, right? And then you've got protons, a whole lot up here, a high concentration up here, and then a low concentration here. So because the lipid bilayer is hydrophobic, because it is nonpolar, hydrogens can't just easily slip on through, right? But they really want to move from this side of the membrane to this to reach equilibrium. But right now, they can't. So a proton gradient, again, unequal distribution of protons. It's divided by a phospholipid membrane. Not only do you have differences in H plus concentration, but you're going to have differences in charge. So while we are looking at positive charges on both sides, this side, the top here, is more positive than the lower side. So while it is not actually negative, it is negative relative to the top, right? It is less positive. All right. And this is all going to influence the movement of protons. So movement of electrons, our friends from the previous slide, is what creates, so through things called electron transport chains, which are a series of proteins and proton pumps, those electron transport chains are going to power the movement of H plus ions to a high concentration to build up a proton gradient. And these proton gradients store potential energy, right? It's kind of like, you want to think of it like a dam. So the membrane is the physical dam, all right? And then we are we're holding back water. It's not actually water, but in our dam analogy, okay? We're holding this back, but it really wants to come to the other side, but it can't. So we're building up potential energy here on this side of the membrane, on the high concentration side of the gradient. And then the controlled flow of protons from one side to the other, which we'll get into later when we look at ATP synthase, is what is going, that controlled flow can be coupled to power cell work, which again, it's why electrons and protons and hydrogen matters so much. So, I told you ATP synthase was coming. Proton flow is going to help produce ATP. Now, let me show you my model here. I've got, again, my high concentration of H plus ions up here. I've got a low concentration of H plus down here, relatively. Like I don't have any H plus down here. All right, then we have our hydrophobic lipid bilayer that's blocking the hydrogen from just passing through. But here in the middle is ATP synthase. ATP synthase is a protein that kind of acts like a turbine in our dam analogy, right? So it actually allows protons to flow through it. As it does, it rotates, and that change from potential energy to kinetic energy, rotating the bottom of this, is actually going to power the bringing together of ADP, which is adenosine diphosphate, and inorganic phosphate to produce ATP, which is our energy transfer molecule. So with this whole setup, which you're going to see in cellular respiration and in photosynthesis, okay, yes, ATP is made in photosynthesis as well. This allows, again, we're looking at energy transfer, right? So here we have potential energy. Then as these flow through, they want to move from an area of high to low concentration, but they need a protein to do it. So they flow through this ATP synthase. Potential energy is changing into kinetic energy, and then kinetic energy is being coupled to this reaction to form chemical energy in the form of ATP. We call this chemiosmosis. It's not osmosis because it's not actual water, but it's chemiosmosis, chemical osmosis, something that's not water, but it's doing the same thing. It's flowing from an area of high concentration to low concentration, and in doing so, driving another reaction. So where am I going to see this throughout the year in AP Biology? Well, like I said earlier, you're going to see this in cellular respiration, because right, the whole point of cellular respiration is to take one molecule of glucose and turn it into way more ATP. So to make ATP, you need ATP synthase, and that whole electron transport chain, proton gradient, ATP synthase situation. In photosynthesis, in the light reactions, which is the first part of photosynthesis, you also need to make ATP to then let the second part, the carbon fixation part, like actually building the glucose, to have the energy to build the glucose from CO2 and water. So you're gonna see it in both of these processes. And then you need to remember, what's the important part of electrons and protons? Electrons tell us about the redox reactions and the flow of energy. So redox reactions that you're going to see, not only, so in photosynthesis, let's just say, we need, we got to produce ATP, but we're also producing those electron carrier molecules like NADPH that you saw at the beginning of this video. And to make those, we need a redox reaction. We're giving electrons from one place to the electron carrying molecule. Then that electron carrying molecule is taking it to another part of the glucose making process. And then it is going to oxidize and give away its electrons to drive that process. Then protons, remember, they help us understand gradients, proton gradients, and chemiosmosis. So understanding hydrogen, understanding the role of protons and electrons, both individually and collectively in AP biology, helps us connect molecular chemistry, which there's a lot of in AP bio, to cell level energy transformations, like how we turn the sunlight, light energy, into chemical energy in glucose, and then how we turn this glucose energy into usable energy in the form of ATP. So all of that is explained just by understanding how protons and electrons move throughout the cell. So now let's see, fluency check, if we get what's really happening. So one way that you can check yourself, because it's one thing to know or to be able to like, oh yeah, I can follow the flow of the electrons. To know if you're fluent in understanding all this at a level that AP Biology requires of you, you want to be able to answer a question like this. So given a model, a drawing or diagram is a 2D model. Could you answer a question like this? A chemical makes the inner mitochondrial membrane permeable to protons. Predict the effect on ATP production and justify using the model. So that's the kind of question ATP is going to ask you. So first, this model is oriented differently than the one earlier, but it's the same model. You've got an area with a high concentration of protons. You have a lipid bilayer, an ATP synthase, even though it's not labeled, like almost all ATP synthases look something like this, because that's how it looks in real life. Sort of. And then you've got an area of low proton concentration, right? So we know that molecules move from areas of high to low concentration. So if we had to draw we want to think, first of all, like, where is the area of high? Where is the area of low concentration? Where are the protons going to flow, right? So normally in this model, protons will move from this side to this side. And man, AP Bio loves to do that, to take a regular picture that, like, they know every single textbook has it turned, right, with the high at the top and the low at the bottom. And they like to turn it some other direction and see if you can still use it to explain what's happening. But this question asks not how does ATP synthase work. It says if there's a chemical that makes the membrane permeable to H+, so if we, like opened up all this, well, predict the effect on ATP production. Now, a very low-level basic answer would be there would be less ATP. But there's no justification, and we're not using the model. So if hydrogen could just diffuse across the membrane without any problems, then it's like there's no barrier, there's no dam. We couldn't build up the proton gradient over here. And without the gradient, we don't have the potential energy. And without the potential energy, we're not going to have the energy transfer from a high potential energy to then a kinetic energy to then the chemical energy that is made when ATP is produced on this side of the model, or on this side of the ATP synthase. So you want to say that, so the effect on ATP production, yes, it's that less ATP would be produced, because if the bilayer, the membrane isn't there, then the gradient cannot build up, and that transfer of energy is not going to happen, resulting in less ATP. So you want to be able to justify, like, step by step, why getting rid of this means less ATP. So you have to walk through all the steps. Give yourself 5 minutes, 10 minutes to try and actually write that out in words for yourself. Like, so now really fluency check is not just do I understand it, can I communicate it in writing in a way that would earn me points on an FRQ. So give it a try and let me know how it goes in the comments. If you found this video helpful, please do give it a like, subscribe to my channel, Miss Parrot Science. I've got a whole lot more chemistry fundamentals, chemistry foundations for AP biology videos coming here at the end of the summer and the beginning of the school year. If you have any comments, questions, or if you just want to leave an emoji down below, maybe a lightning bolt for energy, that would be great. And then you can check out, if you're a teacher, check out, or a student, anybody can buy on TPT. Check out the TPT resources at Know and Grow with Gnomes. I have tons of things on quantitative skills and things that go with the formula and equation sheet, and I'm working right now on building up a bunch of resources that are going to go with this chemistry foundation series. Keep learning, keep growing, and we'll see you next time.